Hi, this is a lecture for my microbes and their human host students. This lecture is over control of microorganisms. So more specifically, we're going to be looking at antimicrobial drugs. So an important quality of an antimicrobial drug or medicine is selective toxicity. Selective toxicity meaning that it selectively kills or inhibits the growth of certain microbial targets, and at the same time, minimal or no harm to the host. Most antimicrobial drugs or medicines currently on the market are antibacterial because the prokaryotic cell provides a variety of different targets. Now, antimicrobial assumes that it's just microbial. So an antimicrobial medicine can be antiparasitic, antifungal, antiviral, and also antibacterial. So today we're going to focus in on antibacterial, but we typically don't call it antibacterial, we call them antibiotics. Okay, there are 5 mechanisms or modes of action of antibiotics. Let's think about this. If you were in charge of developing an antibiotic, you would first decide on a target. A target, what part of that prokaryotic cell do you want to target? Thinking of all the things that you've learned so far about prokaryotic cells like bacteria, if you were in, if you were charged with coming up with an antibiotic, what part of it would you target? They typically just have one target. Um, and then would that target kill the cell? Because that's what the goal is, right? To kill that cell, drastically reduce the numbers. So there are 5. The 5 mechanisms of action of antibiotics. You have inhibition of protein synthesis, inhibition of nucleic acid synthesis, inhibition of cell wall synthesis, disruption of cell membranes, and interference with metabolic processes. So that's how we're going to address these. Of these differences, we're going to look at the antibiotics that are within each of these mechanisms. The first mechanism of action that we're going to talk about is inhibition of cell wall synthesis. In the bacteria, in their cell walls, they contain peptidoglycan. You've learned about this molecule before. Peptidoglycan is unique to bacteria. That means it's in bacteria, but we don't make it in our cells. So it seems like that is a good target. Now, antibiotics that work in this manner, they target different parts of the cell wall. So the one that we're going to talk about is the one that uses beta-lactam rings. So there's a few different ways that antibiotics target the cell wall synthesis. We are only going to talk about one way. I'm only going to hold you responsible for knowing one way, and that is the antibiotics that produce or have beta-lactam rings. All right, so if you look at the picture above me, there are 4 types of antibiotics there: penicillin, empineme, cephalosporin, and clavulanate. And all 4 of those use beta-lactam rings. So beta-lactam rings, you can look at the purple arrows and they're showing you where those beta-lactam rings are. So those types of antibiotics are going to use a beta-lactam ring, and they're going to insert it into the peptidoglycan layer that makes up the cell wall of the bacteria. So as the bacteria are dividing and growing in order to divide and laying down new cell walls, then if the antibiotic is available, if it's there, it's going to lay down beta-lactam rings within the peptidoglycan layer. Now, why is this a big deal? If you can imagine that the peptidoglycan layer in the cell wall is like concrete, And the beta-lactam rings would be like big bubbles in the concrete. As you can imagine, the more bubbles that you have in that concrete, the less stable that it is. And so that's what happens with this type of antibiotic. So if the antibiotic is available, instead of laying down this peptidoglycan, it's going to lay down lots of bubbles. And the more bubbles it has, the more beta-lactam rings in the peptidoglycan, it makes the cell wall flimsy. It makes it flimsy to where it can actually break apart and the cell can die. So of the 4 types of antibiotics that work in this manner are penicillin, emponium, cephalosporin, and clavulanate. Seems great. We've all heard of penicillin. Penicillin was one of the first antibiotics that we had, saved lots of lives, still does. However, there are some bacteria that can counteract these types of antibiotics. And what they do is they produce an enzyme that's going to break down the beta-lactam. So the enzyme that some bacteria produce are called, or the enzyme is called beta-lactamase. Makes sense? ASE meaning enzyme. So beta-lactamase. So if you are treating an infection, let's say with penicillin, that's caused from Staphylococcus epidermidis, it's not going to work because Staphylococcus epidermidis produces the enzyme that breaks down the beta-lactam. So even if that antibiotic is there trying to work, it's not going to. If Staphylococcus epidermidis produces that enzyme that breaks down the beta-lactam rings, that means Staph epi is going to create its cell wall with complete peptidoglycan. Okay, very strong, sturdy wall with peptidoglycan. It's not going to have any effect on it whatsoever. So Staphylococcus epidermidis is one of them that produces beta-lactamase. Some others are Pseudomonas aeruginosa, Klebsiella pneumonia, and Escherichia coli. So if an infection is caused by any of those, and it's given this type of antibiotic that targets beta-lactam, or that has beta-lactam and targets the cell wall, it's not going to work because they produce beta-lactamase. It's just going to break down the antibiotic. So I said this was one way in which antibiotics will inhibit cell wall synthesis. There are other ways as well. And some other types of antibiotics that work in a slightly different way, but still inhibit cell wall synthesis are vancomycin, you may have heard of that one, and bacitracin. All right, so another mechanism of action of antibiotics is to inhibit protein synthesis. So to inhibit protein synthesis in the bacteria. And it does this by preventing translation. So the antibiotic is going to bind to the ribosomes and prevent translation. You guys know all about protein synthesis. Remember, protein synthesis includes transcription, then translation. It occurs in our cells and it also occurs in bacteria. So this type of antibiotic will bind to the ribosomes, the bacterial ribosomes, and prevent translation, prevent them from doing their jobs. Now, at the very top picture, you see an example of a bacterial ribosome. You know that ribosomes, when they're not translating, are in 2 different parts. We have the small subunit and a large subunit. When they come together, that's when they undergo translation. And they will translate a messenger RNA strand. Now, you've also learned that bacterial ribosomes are slightly different than eukaryotic ribosomes. But there was that one exception to the mitochondria, right? The mitochondrion have ribosomes that are very similar to bacteria. Okay, but our regular mitochondria that we have in our cytoplasm is different than the bacterial ribosomes, slightly different. It's different enough that the antibiotics that target, where their target is to target protein synthesis, it will have no effect on our cytoplasmic ribosomes. Okay, so it's going to prevent translation. How is it going to do that? If you can look at the other picture on the slide, yep, this one right here, it's showing you tetracycline. Tetracycline is an example of an antibiotic that works in this manner. So tetracycline is attaching to the small subunit. If it attaches to the small subunit, the large unit may not be able to bind, and so it prevents translation. So just as simple as that, these antibiotics are going to bind to either the small subunit or the large subunit and won't allow them to undergo translation. So some antibiotics that work in this manner are tetracycline, streptomycin, gentamicin, tobramycin, doxycycline, erythromycin, clindamycin, linezolid, and dalfopristin. Those are all that I have listed over here on your slide. Now, I'm not going to hold you responsible for memorizing all of these and how to spell them. What I would like you to know, I would like you to be able to recognize them. Be able to recognize them as being antibiotics that inhibit protein synthesis. Now, there is one I want to talk about kind of as an aside, give you a little bit more information about, and that one is tetracycline. That is the first one on your list and the one that's given as an example here in that picture. Tetracycline, what we know now, when tetracycline was first developed and people started to take tetracycline, worked just fine, but it had a side effect to it. What they started to notice is that there were some babies that were born that had stained teeth, and it was on their adult teeth as well. What they figured out was that it was a side effect of the antibiotics. So the mother, when the mother was pregnant with the baby and they took tetracycline, it actually formed a complex with calcium. So when the fetus was developing the teeth, when it was laying down the calcium into the teeth, it also laid down the tetracycline. And so the babies were born with teeth that had the staining effect from this tetracycline. There is no way to take that tetracycline out after it was laid down. So many people were born with the stained teeth because of the effect of that tetracycline. They didn't figure that out until many years later. At the time when they developed this, they weren't as good as we are now at looking at side effects, okay, and discovering the side effects and knowing what they were. We have advanced in light years since then. So there are still some people out there that have to deal with those discolored teeth because of that. And that's tetracycline. Okay, another mode or mechanism of action is to inhibit nucleic acid synthesis. So let's not forget what we're talking about. We're talking about how antibiotics work, what they're doing, what their target is in the bacteria. So this type of antibiotic inhibits nucleic acid synthesis in the bacteria. Because bacteria have to synthesize DNA, okay, nucleic acid, they have to synthesize nucleic acid. They also synthesize RNA. So they're going to synthesize DNA whenever they're going to go through binary fission. They're going to synthesize RNA whenever they're going to make proteins. So this type of antibiotic inhibits nucleic acid synthesis, prevents the bacteria from making copies of DNA and RNA. Now, when bacteria need to make DNA and RNA, they use enzymes for that. So the antibiotic will target the enzymes required to make DNA and RNA. There are two enzymes that I would like you to know, and that is DNA gyrase and RNA polymerase. So there are antibiotics that will target DNA gyrase and RNA polymerase. They will deactivate those enzymes, or they bind to the enzymes. They're going to block the enzymes so the enzymes can't work. And if the enzymes can't work, that cell cannot make DNA, so it can't replicate. If RNA polymerase doesn't work, it's not going to make RNA. Therefore, it's not going to make proteins that it needs to stay alive, so it kills the cell. Examples of antibiotics that work in this manner, I have listed on your slide. So ciprofloxacin. Okay, the next one I really have trouble with pronouncing all the time. It's called metronidazole. It's my best. And then the third one, rifampin. And I have a kind of another aside that I talk about as far as rifampin goes, which it's kind of like an interesting tidbit about rifampin. Rifampin, when someone is given rifampin as a side effect, and it's not a toxic side effect at all, but it will color body fluids. So it will color it a reddish to an orangish color. Now, it's not toxic at all, and it doesn't stay. It only stays as long as the antibiotic is in the body. But if you think about this, if you're given rifampin, your saliva, the sweat, sometimes your feces, your urine will all take on this reddish to orange to brownish color. And that can be a little alarming. So if you're on rifampin and your urine turns a reddish brown color, yeah, that can be alarming. Or if your tears are a reddish brown color, that can be alarming. If you wear contact lenses, it could stain those contact lenses. So just to one thing kind of to be aware, that's just the side effect of rifampin. It's not long-term or anything like that. And it's not toxic. All right, our next one, mechanism of action of antibiotics is to interfere with metabolic pathways. So thinking of all of the metabolic pathways that could be going on inside of that bacterial cell, there's a lot of them. But if you were in charge of making an antibiotic and they said you have to make an antibiotic to interfere with metabolic pathways, any metabolic pathway. So then you'd sit down and maybe you'd make a list of all of the metabolic pathways that occurred inside of bacterial cells. And if you were doing that, you'd probably come across a lot of metabolic pathways that also occur in our own cells and eukaryotic cells. And you think, well, gosh, I don't think I want to develop a medicine that's going to interfere with a metabolic pathway that's also occurring in my cells, because then that person's going to have a lot of side effects because of that. You're going to look at metabolic pathways that would only occur in bacteria and not occur in eukaryotic cells. That would make more sense. So I'm going to give you one example. Most of the antibiotics that fall within this category interfere with folate production. Okay, so folate. If you've heard of that word before, folate, another word for that is folic acid. You probably heard of folic acid. If you look on your maybe a multivitamin that you take, folic acid sometimes is in there. Now, here's the thing. Bacteria make folic acid, but our cells do not make folic acid. So that seems like a win. This is a metabolic pathway that occurs in bacteria that does not occur in our cells. So within our cells, we do not make folic acid. We don't make folate. We actually have to get but our cells need folate. So we have to get folate from external sources. Where we usually get folate is from our gut bacteria, the bacteria that is living in our large intestine. And we can also get it from other sources, but our main source is from the bacteria living in our intestines. All right, so now we do have a metabolic pathway that is occurring in bacteria and not in our own cells. And so these antibiotics are designed to interfere with the process that the bacteria use to make folic acid. So if you look at the image that I have for you, and let's look at the very top green box. So if in the bacteria you're going to make folic acid, you have to have a starting molecule. You're going to undergo some chemical reactions to get another molecule. Sometimes you undergo another chemical reaction to get another molecule in the process of making folic acid. So let's look at this, says precursor number 1. And we said that it undergoes some chemical reactions in order to get to the next step. Those chemical reactions rely on an enzyme, which they do. There are some antibiotics that will target that enzyme. So it will destroy the enzyme. If the enzyme is not there, it's not going to be able to make the folate. So it interrupts that folate production very early on in the production. Okay, so sulfa drugs, that's what they call them. It's an earlier type of antibiotic. Sulfonylamides are also called sulfa drugs, type of antibiotic. They will destroy the enzyme that's needed early on in the process. There's another type of antibiotic that interferes with folate production, and that one is called trimethoprim. So look at the bottom arrow, trimethoprim. It destroys a different type of enzyme, but also an enzyme that is responsible for folate production. So it's later on in the production of the folate. But either way, if you're destroying an enzyme that is needed in order to make folate inside of that cell, that bacterial cell is not going to make it. And if the bacterial cell cannot make folate, then that bacterial cell cannot stay alive, because bacterial cells also need folate. So they need folate to stay alive, and if they're not making it, they die. So these types of antibiotics are interfering with the metabolic pathway of folate. Okay, the last type of mechanism of action of antibiotics are to interfere with cell membranes. So interfere with cell membranes. Think cytoplasmic membrane. Now we know that bacteria have cytoplasmic membranes. We've already established that. And what happens with this type of antibiotic is that the target is going to be the cell membrane, the cytoplasmic membrane. Now, the very top picture shows you that phospholipid bilayer, shows you a cytoplasmic membrane. You see the integral proteins, you see the other molecules associated with a cell membrane or cytoplasmic membrane. And We also know that our cells have cell membranes. We know that bacterial cytoplasmic membranes and human cell membranes are almost identical. So this type of antibiotic is going to target those cell membranes. Now, the second picture I have, the one that's closest to me, right up here, I like this picture because it shows that phospholipid bilayer and it shows it on a microbe. So this is the phospholipid bilayer on the microbe, the cell membrane on the microbe. And what the antibiotic does is basically it pokes holes in the membrane. So it's going to poke hole in the membrane and get the membrane to get the cytosol to leak out, and therefore the cell dies. Anytime you're going to poke a hole in a cytoplasmic membrane of a bacteria, that cell is going to die. It doesn't reheal itself, it's just going to die. So there are two types of antibiotics that I would like you to be able to recognize that work in this manner. It's daptomycin. Daptomycin really works against gram-positives. Okay, it's really good at working against gram-positives. Another one is called polymyxin B, and it's really good at working against gram-negatives. Now, most of the antibiotics that fall within this category are topical. So do you know what topical application means? It means that you're going to use it on the surface of your skin. You're not going to take it orally or by IV. You're not going to take it internally. It's going to be topical. So like Neosporin type is what I'm going for topical. Now, why? Why do you think that is? Why do you think that most of the antibiotics within this category are topical and not, you wouldn't take them internally? Well, if they're going to interfere with the cytoplasmic membrane of bacteria, our cell membrane is almost identical. So what it's going to do, it's going to start destroying our own cells, cytoplasmic, our own cells, cell membranes. And that's what we don't want. So for the most part, it is topical. On certain occasions, your physicians might use this as an oral. It's very limited on certain conditions because if it is taken internally and not watched very closely, it can lead to kidney and liver damage. It's going to hurt the kidneys first, and then it'll hit the liver, and it could cause those organs to shut down. So it's very rarely used as an internal medication, only on topical. So the next time that you do use Neosporin or a triple antibiotic that you buy like at the store, read on the little tube and read to see which types of antibiotics are on there, right? You're going to probably see daptomycin, polymyxin B, and maybe another one. All right, so that is it. You learned about all of the 5 types of mechanisms of action of antibiotics, and they're all against bacteria, right? These are all antibacterial medications.